Hello students, today we are going to learn about the immunity, different type of immunity, right? Now here immunity, first question is immunity. Immunity, every day we are exposed to a large number of infectious agents. Every day, because we know pathogens, they are present in air, water, soil, everywhere pathogens are there. Pathogen means microbes are present everywhere. Bacteria, they are present. So we are also face all these microbes every day. Is given here, every day we are exposed to a large number of infectious agents. However, only a few of them exposures result in disease. Only a few that causes for disease. Question is why? Because answer is also given. This is due to the fact that the body is able to defend itself from most of these foreign pathogens or agents, right? The ability, what is given? The overall ability of the host to fight with pathogens or microbes or disease causing organisms confirmed by the immune system, and this is called immunity. This is called immunity, and also given here immunity there are two types, innate and acquired immunity. Innate means immunity which we get by birth, right? And acquired means which we get after birth. Okay, so first we are going to read about the different types of immunity. Before going to read this NCERT, first I tell you something about the immunity. First, what is given here? Immunity, right? So first means what? Immunity. What is immunity? First question is here, what is immunity? Immunity means what? Immunity means what? It is the resistance property of our body which fight against the diseases. Simply we can say resistance against disease or disease causing agents, pathogens. In very short term, we can say what is immunity? It is the resistance property of our body through which we fight with the disease or with the pathogens, right? Another term is also come in our immunity, and that is mostly antigen, pathogen. We use pathogen, and pathogen they contains antigen. So here question is what is antigens? Another word, it is antigen. Antigen is also called as here agglutinogens. They are also called as agglutinogens, right? Antigen or agglutinogens. These are the substances which are present on the surface of the pathogen. These are substances which are present on the surface of the pathogen, right? And they induced or they stimulate the secretion of antibodies, right? We can say here these are substances which stimulates the secretion of antibodies. First thing is that, but where they are present? They are present. Present, present on the cell membrane. Cell membrane of pathogens, we can say cell membrane of pathogens. We also know in our blood cells, RBC cells, they contain these antigens, polysaccharides, glycoproteins. They are glycoproteins, right? They are antigens which are present on the cell membrane, and they are responsive to stimulate the secretion of antibodies. Okay, now antigen is given here. So another term is also we have antibody or antibodies. Antibodies, they are also called as like agglutinogens. They are proteins. So we can say they are agglutinin, also called as agglutinin, right? Antibodies means what? They are proteins, army of proteins which stimulates due to the action of antigens, which secretes or stimulates, which are secreted by lymphocytes B due to the presence of antigen. These are antibodies. They are proteins. We will read the structure of antibodies in this today's lecture also, right? Now next point is here, that is here to understand antiserum. What is antiserum? When I tell you at the time of when I teach you about the blood, that time I tell you serum and plasma, right? So what is serum? What is the difference between serum and plasma? That time I tell you means a plasma, or we can say matrix, in which clotting factors are absent. This one is called serum, right? So means we can say plasma minus clotting factor is called serum. So what is antiserum? Antiserum means if matrix means plasma, we can say, right? That is plasma with here we can say matrix, blood plasma. You understand why I write here plasma? This plasma means it has no blood coagulating substances, right? Blood coagulating agents and blood cells are absent here. So blood plasma with antibodies of others means other organisms. These are considered as antiserum. Means these serum means these are plasma matrix, and they are not from the same organisms, but it is come from the another organism, right? Another animal or another organisms, and they have antibodies. So with antibodies, blood plasma with antibodies called antiserum, right? Venom means it is toxin. I did not do it here. Serology here. One term which is also important, because here we are talking about the antigen and antibody. Here in study of interaction between whom? Antigen and antibody. The study of interaction between antigen and antibody is called serology, and this process is called which process? Interaction between antigen and antibody process is called agglutination, called as agglutination. Simply we can say agglutination means what? A study of agglutination is called serology. A study of interaction between antigen and antibody is called serology. Sero comes from serum. Okay. Now immunity, different type of immunity. Here types of immunity. Types of immunity. What are the types of immunity? When we consider the types of immunity, now we have first two types of immunity. Some immunity we get before birth, right? From the embryonic stage, and some immunity we acquired after the birth, right? So here two types of immunity is there, right? The first one is here we can say this immunity, it is innate immunity. Innate immunity, right? And second is our acquired immunity. Acquired immunity. So now we have two types of immunity, innate and acquired. Acquired, innate means what? It comes by birth. And this is acquired means after birth. Through atmosphere we acquire. Now here innate, when we consider the innate immunity means it is not specific. What are the means we can say what are the specific feature of innate immunity? Innate immunity means it is non-specific. This one is non-specific. Non-specific. This one is non-specific, right? But acquired immunity is species specific. Species specific. It also contains memory, right? Memory. Third characteristic of this acquired immunity, it is discrimination between self, non-self, self and non-self, right? And these are absent here. It cannot means the innate immunity, in innate immunity it is absent, right? In acquired immunity this one is present. Now next here, memory, species specific memory, and one is also there. It shows also diversity. Diversity means different type. Species specific means how many types of antibodies are there? We simply said this much. It is according to the number of infected against. How many different species of pathogens they affect us, and for it is specific pathogen, specific antibodies formed. So different type of diversity. So these are four properties or characteristics of acquired immunity, right? And innate immunity means here barriers are there. It contains barriers. Which barriers? Now here when we consider barriers, so we have four types of barriers. How many? 4 types or 4 barriers are there, right? And these here we can say first barrier is our physical barrier. Right? Physical barrier means here you understand physical barrier, it contains skin and mucus or mucus, we can say mucosa layer or mucosa, right? So these two consider as physical barrier, skin and mucosa, right? Skin means what? Skin, look here, skin, it is a stratum corneum, actually our skin means here, okay, I will describe you detail, right? First here only different type of immunity we will read this, right? Then after one by one I tell you. Skin and mucosa, right? Physical barrier. Mucosa means it is the internal organs lining of specific, that is our tract. Which major tracts are there? Gastric, that is respiratory tract, gastric tract and urinogenital tract. So lining of respiratory tract, lining of digestive tract, lining of urinogenital tract, right? It contains mucous membranes are there. This means pathogens, they are stuck into the mucus, and mucus contains phagocytes. They eat and destroy this one. Another thing is there, they are ciliary movement are also there, right? In our as for example in respiratory tract, ciliary movement is there. So ciliary movement means what? They propel this mucus toward the outer side simply, right? And skin means our skin, we know our skin is a which type? It is a keratin stratoepithelium. It is, we can say it is keratinized stratified epithelium. What happens? The outermost layer of our skin, it is made up of keratin, means dead proteins, and also pH of skin is acidic, right? Second barrier is our physiological barrier. Right? Our physiology like pH in stomach, that is HCl, fever. Here some examples are, as for example, fever, pH, right? pH value, of skin and some secretions. Third is our cellular barriers. Cellular barrier. So cellular barrier means it contains cells. Which cells? Our WBCs. Which WBCs? These are called as P, M, and L. Phagocyte is here to process is there, phagocytosis, right? And natural killer cells are there. Monocytes and killer cells. Natural killer cells. Lymphocyte, natural killer cells. And fourth barrier is here our cytokines. Cytokines. And cytokines, it is present only in virus infected or cancer cells. As for example here, alpha interferon. It is secreted by, this one is secreted by the cancer cells or virus infected cell. Actually, alpha interferon, right, it is secreted and it helps to protect the other cells, because other cells, they, what happens, they forms TIP. TIP means what? TIP, translation inhibiting proteins. And when translation inhibiting protein happens, then what happens? The retrovirus process is stopped. Formation of virus. So they can inhibit the translation process, and through this translation process, they also protect themselves from viral infection, or we can say from cancer cells, right? So these are four barriers. And also when we consider the line of defense, so we have three line of defenses in case of our innate immunity. Three line of defense means what? What the three line of defense? The first line of defense is physical defense. Physical defense. Physical means here what comes? That is physical, skin, mucosa, and physiological barrier. They come into the first line of defense, right? Second line of defense is biological defense. So biological defense means what? Cellular. So cellular barrier, it comes to the second line of defense, right? And third line of defense means what? Acute immunity, right? Acute immunity means that is lymphocyte. So lymphocyte T, lymphocyte B, when they form, that means antibodies, lymphocyte T, lymphocyte B, they are come into the third line of defense. That is called acute defense. You understand? When we consider the line of defense, we have three types of line of defense. Here I write, then you understand. When we consider three lines of defense, line of how many? Three. Defense. So when we consider line of defense, how many lines are there? Three lines. Three lines. Which one? First is called physical defense. It is also called as first line of defense. That is first line of defense, right? Second one is our cellular defense. Cellular defense. Cellular defense means it is the second line of defense. And third one is our acute defense. Acute defense. Acute defense means here it is our third line of defense. And here what happens? Here lymphocyte T or B and monocytes, they are involved here in acute defense. So we have three line of defense are also there, right? First line of defense means physical defense, in which physical and physiological barrier comes. Second line of defense, cellular. Cellular means here WBCs. Which one? That is PMNL, monocytes, natural killer cells, means they are coming, right? Last is our acute defense. Acute defense means they are memory based. We can say here third line, lymphocyte T, lymphocyte B, that is cellular mediated immunity or antibody mediated immunity comes into the acute defense. That is our third line of defense. Antibodies, they are also come into the third line of defense. You understand? Okay. So these are some, we can say, these some important information about the innate immunity. Now second one is our acquired immunity, right? So first I only divide this. I only inform you about the different type of acquired immunity. Then I start from one-to-one immunities, right? Okay. Now second is our acquired immunity. Acquired means we get this immunity after birth. After birth, right? So we get this immunity after birth. Now acquired immunity means when we consider this one, it is also two types. One is called here, right, active immunity, right, immunity, and second one is called our passive immunity. Active and passive immunity, right? Now active immunity, here our immune system responses, but here what happens, here immune system is not active, right? Active immunity means we have to induce pathogens inside the, or means pathogen proteins. Pathogen protein means it is antigen, right? When we induce or introduce pathogens in our body, then what happens, our immunity, it also gives a response, right? When our immunity is active, then it is called active immunity. Active immunity is also further divided into two types. Which one? It has two types, and now it is also two types. First one is natural, and second one is artificial. Natural and artificial. When we consider, as for example, for natural immunity, what are the example? Simply, what we get through nature. Nature means from nature. Nature means what here? We can say infection. Through infection, right? So different pathogens, they infect us, and when they infect us, then what happens? Our this immunity, it becomes active and forms which response. That is called our infection, active immunity, natural active immunity. Second is our artificial immunity. Artificial means by man, right? So here what happens, when we inject weak or live or dead pathogens in our body, or we introduce proteins, that is antigen, proteins of or antigen of this this infectious agent, then what happens? Our body get responses from, give responses. Vaccination, through vaccination, right, or antigen, right, when these are injected in our body. So through introduce, vaccination is artificial, because human, what happens, we introduce some pathogens, that is weakened pathogens, right, into our body, and then what happens, our immunity give us some response, right. Second is our passive. Passive is also same as to this one, two types. One is, we can say, natural, and another one is our artificial. Natural and artificial, right? Natural and artificial. So natural immunity, for nature, passive immunity means we get from nature, right? So this is, we can say, antibodies which we get from our mother. We have two types. Two examples are there. Placenta. Through placenta, which antibody we get? It is IgG. And second is our colostrum. Through colostrum we get IgA. These are antibodies which we get through natural, from mother, right? These are examples. And artificial means, here I can write here anti-serum, which I already tell you what is anti-serum, right? Means we can say ready-made antibodies are introduced. Made antibodies, they are introduced in our body, right? Means here what happens, in both of these, already ready-made antibodies are introduced in our body naturally through placenta in embryonic stage, or colostrum postnatal stage. Artificial means when these are the already ready-made, means they are already performed or ready-made antibodies which are introduced in our body. As for example, that is a tetanus, right? Tetanus. In case of tetanus, what happens? Tetanus means Clostridium tetani. Then what happens? We get anti-serum at that case. Or in some case of venom, snake poison, right? We get ready-made antibodies, right? So these are the different types of acquired immunity. Okay, now we start from our innate immunity. A little information I will tell you here, not very deep, right? And which is enough for NEET exam. The first is innate immunity. Non-specific, and we get innate immunity by birth. Already I tell you. Innate immunity means how many barriers are there? Here I am talking about the 4 barriers. 4 barriers, right? Which barrier? First one is our first one is your physical barrier. Physical barrier, right? Physical, simply you understand physical means 2 types of barriers. 2 types. One is our skin and second one is our mucosa. Skin and mucosa. Skin and mucosa. Skin means it has keratinized stratified squamous epithelium, right? So this keratinized squamous epithelium means we can say the outermost layer of our dermis, epidermis, right, which is made up of, that is we can say it is stratum corneum, right? So stratum corneum, it has dead cells proteins. So in that means it has also pH, is acidic pH. So what happens? The bacteria or infection cannot be happen here. Okay, you write, then I tell you, right? Which it is stratum corneum. Stratum corneum, outermost layer, right? Which is made up of dead cells, dead cell protein, right? It prevent the entrance of microbes, and also the pH of skin is also here. We can say pH of skin is 3 to 5, right? So 3 to 5 is what? Acidic, due to the acidic, under acidic pH. So what happens? Bacteria cannot grow on skin or they cannot enter into the skin. Second is our mucosa. I write it just below the mucosa, then you understand, right? Second is our mucosa. Mucosa, it is present where? In the lining of, where they are present? In the lining of major tracts. Major tracts. As for example, in which one? Respiratory tract, gastrointestinal tract, and urinogenital tract, right? They are present. What happens? Mucosa already I tell you, they trap the pathogens, which we suppose that we, what in respiration what happens? We inhale the mixed air. Through this mixed air, spores or bacteria, they are also inhaled this mixed air. When they reach into in the respiratory tract, what happens? This respiratory tract is lining by the mucosa, and this mucosa layer means it secretes mucus. So they are trapped into the mucus, and when they are trapped into the mucus, then what happens? In mucus, also phagocytes are there, so they engulf these bacteria and digest. Second process is there, these mucosa means as well as, for example, in respiratory tract, what is there? There we know here ciliary epithelium is there. So these cilia, they propelled the mucosa outside toward the mouth side, right? And this mucosa, which is contaminated with the pathogens, it comes into the mouth, and from mouth it goes outside the side. So this mucosa also act as physical barrier. Now second is our physiological barrier. Physiological barrier. What is there? It is physiology. What is given? Physiology. Physiology means some process which are going on in our body. As for example here, first here three types. One is our fever. Fever is not a disease. Fever is not a disease. Why I tell here? Why I write here? Fever is not a disease. Fever means what happens when our body is infected with any other type of pathogens, or we can say a different type of protein is present in our body. Some toxic substance are present in body. As already we read in malaria, what we read? Hemogyne. When hemogyne is present in our blood, then what happens? Our homeostasis organ, that is our hypothalamus, it increases the body temperature. When it increases the body temperature, that process is called fever. When body temperature increase means fever. So this is a physiological barrier. So in fever, what happens when body temperature increase? Then what happens? It stuck the reproductive growth of bacteria, or it checks the fever means it the multiplication of pathogens, right? Second is there our, that means it is a cross of immunity. Second is here pH value. The pH value of our internal organs or tract. As for example, in gastric juice, what is there? pH. HCl is released. HCl, and you know HCl means what? Its pH is 1 to 2. Acidic medium, right? Highly acidic. Okay, when you consider the urinogenital tract also, urine is also acidic. Urine is acidic. That's why it also protect from the bacterial infections, right? In case of vagina, you also their lactobacillus is present, means acidic. So what they form? They form the acidic medium so that the bacteria cannot grow in these tracts, right? And one more third is here secretion. Secretion. So some secretion, as for example, our eyes, tears, sebum. As for example, in our ear, sebum is there, right? They have also acidic medium. They are also acidic. So they also protect us from infection. They also check the growth of acidic. They protect, right? Protect us from bacterium by stop the multiplication. Stop the growth of pathogen. So this one is your physiological barrier. Third, now when we consider the third one is our, you can write here, right? Okay. Third is our cellular barrier, right? Physical barrier, second is our physiological barrier, and third one is our cellular barrier. Cellular barrier. Cell means here cells are involved. This one is cells are involved. Now here first and second, which are physical and physiological barrier, they come into the first line of defense, and this is the cellular barrier. The cellular barrier, it comes into the second line of defense, In case of cellular barrier, we have different cells. Which cells are involved here? We can say PMNL. PMNL means polymorphonuclear lymphocyte. Polymorphonuclear lymphocyte, right? You understand what is the meaning of poly? Poly means many, morpho means morphology, nucleus means comes from nucleus, right? So different type of morphology of nucleus is present in these. Leukocyte means WBC, and I think you understand different type of different lobes are present here. 2 lobe, 3 lobe, 3 to 5 lobe, or kidney shaped structure. That is, different type of nucleus are present in these lymphocytes. That means here we are talking about which one? We are talking about the granular lymphocyte, granular leukocyte. Leukocyte means WBCs. Second, this is first one. Second, it is monocytes. That monocytes, they are present inside the blood. They are called monocytes. But when they come into the tissue, then they are considered as, in tissue, they are considered as macrophages. Macrophagous cells, right? In tissue. Third is our natural killer cells. NK cells means natural killer cells. They may be lymphocytes. These are lymphocytes, specific lymphocytes. These three types of cells are involved in cellular barriers. How they defend us? And what process they? So they have two processes. First one is called phagocytosis through phagocytosis. Phagocytosis means they eat the foreign part, virus, bacteria, protozoan, or debris also, right? Blood cell debris. Second is eat and digest. Second one is here non-phagocytosis. Non-phagocytosis. So non-phagocytosis means here what happens? They cannot eat the pathogens, but what happens? They make spores in their cell wall, right? In their cell wall or in their cell membrane. And when they make spores, then what happens? Water content enter into the pathogen cells. They swelled up and then blast. That means blast. Blast means burst. And when they burst, then they are converted into the debris. And these debris are eaten up by the phagocytes, right? And thus they clean the blood. Okay, first phagocyte. What process happens in the phagocytes? In case of phagocytosis, what is there? In case of phagocytosis, here we have actually we have 5 stages. So what are the 5 stages in case of phagocytosis? So 5 stages means what? First, at the point of or at the site of infection, what is there? Histamine is released, right? Here, phagocytosis, it contains 5 steps. 5 steps. Which one? First is here dilation. So dilation means what? Here dilation of blood vessels, right? So first here what is there? Here we can say dilation. This dilation means it is vasodilation, because here vasodilation. Vasodilation means why it is also called as vasodilation? Because here blood vessels are dilated. So blood vessels, due to the why they are dilated? Due to the secretion of histamine. Which one? Histamine. This histamine. And this histamine, it is, who secrete this one? Histamine. Who secrete this one? Mast cell. In case of tissue, it is mast cell. and in case these mast cells, if they are present in the blood, then they are called as basophils, right? They secrete the histamine, serotonin. So histamine, it is vasodilator. So what happens? It delayed the blood vessels. Delayed the blood vessels means, look, these are the blood vessels. Now it delates means this site where infection happens. So this side, the vessels are delayed. The vessels are delayed, right? Now this is due to the histamine, right? Here, who secrete mast cell. Then what happens? Now blood flow is there. But here what happens? Here flow is less. So what happens here? Accumulation. Second point is here. First, vasodilation, and second, adhesion. Adhesion, or we can say accumulation. The second process is adhesion. Here, accumulation of, I can write here, accumulation of which one? WBCs. Where? At the site of deletion, at the site of infection. Look, these are WBCs which are accumulated, right? The second is accumulation of WBC due to the slow movement of blood, right? Third is here diapedesis. APEDESIS. Diapedesis. Diapedesis means it is a process in which that WBCs move from blood vessel to tissue or blood vessel to ECM. Here, movement of amoeboid movement. Amoeboid movement of WBCs from blood to ECF. ECF means extracellular fluid, means we can say tissue fluid. Right? In tissue fluid. This movement is called diapedesis. Fourth one is our chemotaxis. Here you can understand, right? A small pores are present here. These small pores, these are WBCs, right? These WBC, they can come out through these pores. So this movement is called diapedesis. This is blood capillary, and blood capillary, you know, only one layer is there, and that is endothelium. So they have some gaps in between, right? So when this vasodilation occurs, these gaps, they can we can say size of gap is also increased, and because they are amoeboid, WBCs are amoeboid in nature, so they can easily come out from this vessel. RBCs never come out of the vessels because they have a specific structure, right? But WBC, they are amoeboid cells, so they can out of the blood. Chemotaxis means what happens? It is chemical movement. Here, chemical means what? A specific antigen which are present in the antigen. That is, actually these antigens are chemicals. So the specific antigen which are present on the pathogens, they attract the WBCs, right? Then we can say chemicals of antigen oblique pathogen, otherwise you confuse this one, pathogens antigen, right? They attracts the WBCs. So WBCs, they show chemotactic movement. Who chemical? These chemicals which are produced by pathogens or the antigen which are present on the pathogens, they attract the WBCs. Now WBC move toward the antigen which are present inside the lymph. And fifth one is our phagocytosis. phagocytosis. So phagocytosis means we know here 3 steps are there. First, what happens? Attachment, adhesion that we can say, then engulfing, and then digestion, right? So 3 things are there in phagocytosis, same as to amoeba, right? Here what happens? First is attachment. Attachment is also called as adhesion, right? Or adhesion. Then what happens? Engulfing. Engulfing means simply they engulf it in this one, right? It is called INGESTION or engulf, right? Then what happens? Digestion, intracellular digestion. Intracellular digestion. Intracellular digestion means inside the cell they digest, right? So this is the phagocytosis. First, what happens? When they attach to the pathogen, that is means substance, then what happens? They form pseudopodia, and these pseudopodia, like amoeba, they engulf this whole pathogen inside the cell, right? Now this specific food vacuole, now a food vacuole is formed, simply this food vacuole is called phagosome. When this food vacuoles it merge with lysosome bodies, then it becomes phagolysosomes or lysophagosomes, right? These phagolysosomes, now what happened? Due to the presence of hydrolytic enzymes in lysosomes, it digests the pathogen. That is called, they are slaughtered inside the cell. After digestion, that what happens? These are clear. So this is called phagocytosis. Second one is there. Second is non-phagocytosis process. Second is non-phagocytosis process. In case of non-phagocytosis process, natural killer cells are used. Natural killer cells are lymphocytes with specific, means we can say perforin proteins are there. Then what happens? They simply, second cellular difference is non-phagocytosis. In case of non-phagocytosis, what I tell you here they did not digest the debris or digest the pathogen. But what happens? These cells, these are natural killer cells. They forms pores in cell wall or cell membrane, or membrane of pathogen. Then what happens? H2O, water, enter in pathogen cells. Then what happens? Swelled up, swelling is there, and then burst. Burst means kill, and convert into debris. Then they convert into debris, and this debris is phagocytosis through phagocytosis through which cells? Macrophage cells, right? Neutrophils or macrophage cells. So this is the non-phagocytosis. They make spores, and then what happens? Through pores, water enter into the pathogenic cell. Now cell swelled up, and then after what happens? It burst, right? And our last, that is fourth barrier, it is cytokine. Cytokines. Here cytokines means what happens? The virus, infected cell, or cancer cells. cancer cells, they secrete a specific protein, that is alpha interferon. They secrete a protein, and this protein, it is made up of how many? 270 amino acids. Protein made up of 270 amino acid. That is called alpha interferon. Alpha interferon. How many? 270 amino acids. Right? 270 amino acids. Alpha interferon. Right? One thing. Second, and due to this interferon, what happens? It protect the neighbor cells. By what? By how? By stimulating TIP. TIP. That TIP means what? TIP means it is translation inhibiting protein. Now you understand translation inhibiting protein. If translation inhibits, then what happens? Proteins are not formed. Translation means what? Formation of protein. If translation inhibit means proteins are not formed. Proteins are not formed, so that means what happens? This means we can say multiplication of virus or multiplication of cells, they are inhibited, right? And so our body protect from cancers or viral infections. The neighbor cells, they are protected through this one, right? Here also this non-phagocytosis is not for only pathogens only. Sometimes what happens, suppose that these are infected cells, or infected cells, they are also digested or they are also killed through this non-phagocytosis, right? So these are 4 barriers which are present in our body, right? Means in our body in the form of innate immunity. These are 4 barriers which come into the innate immunity.